Hello everyone and welcome to Lecture 2 of Unit 1, and today we are going to be discussing gastrointestinal histology, and this is going to include both macro and microscopic anatomy of the gastrointestinal tract. So the first part of the GI tract starts with the mouth, and a very critical thing that needs to occur within the mouth is salivation. So saliva actually comes from the salivary glands, and there are actually three different types of salivary glands. The first type is serosa saliva. So this is a very has a lot of water, a lot of electrolytes. This buffers the rest of the GI tract, especially the stomach, and it comes from the particular salivary gland called the parotid gland. And these parotid salivary glands in cattle and other ruminant species are much more developed than they are in carnivorous animals, for example. The next group is going to be those that produce mucus saliva, and this saliva is very rich in proteins and enzymes. So if you've heard, saliva has amylase and other enzymes involved. So this is mucus saliva. And then the third type is mixed salivary glands, and these salivary glands actually produce both the mucus and the serous saliva. So here we have an example of a dog, a pig, a bovine species, and a horse. And you can see how, based off of what the animal mainly consumes, how these salivary glands differ. So you can see the orange tannish color, those are the parotid glands, and that's where they're at the back of the mouth. They produce the serous, the watery, electrolyte-rich, great buffer saliva. You can see in the herbivorous animals, the cattle and the horse, that those are much more developed when comparing those to the dog, for instance. The yellow is the sublingual gland, and this is the salivary gland that's located under the tongue, and this releases very protein-rich mucus saliva. And then you have the blue, the light blue, as the mandibular gland. This produces a mixed saliva. And the red is the buccal glands. These are more classified as minor salivary glands. They also produce mixed. They're usually more up on the top of the mouth. And those are classified as minor. They change greatly depending on the animal, and there are many different glands throughout the mouth. But those are the main ones that I want you to know for this class. So here's an example just looking at the different ruminant species on how these salivary glands can change very greatly. So on the left-hand side here, you can see the concentrate selectors, which include deer. So these animals are going to be eating, there are browsers, they eat more leaves, compared to on the right-hand side, which includes cattle, antelope, and those are more of the grazers. They mainly are eating grass. So you could see that the concentrate or the browsers, they have much more significant parotid glands compared to the grazers. So you could see here that, for instance, the roe deer, the parotid gland is 0.22% of the body weight of the animal, compared to cattle, where that parotid gland is 0.06% of the body weight of the animal. It enters into the lumen of the salivary gland, or the duct, and then once it's within the duct, you have the saliva is actually modified. So it typically, depending on the type of saliva, we are either adding or removing water or electrolytes. A lot of times we are removing a lot of the electrolytes that are initially released from the secretory cells. So here is a histological picture. So we have both the mandibular gland on the left-hand side here, and you can see this is a mixed compared to a parotoid gland, which is serous. So this is more water electrolytes compared to the protein. You can see here the protein, with this type of staining, protein doesn't really take up that stain. So all these white parts, those are all the proteins within the salivary glands, compared to that of the parotid gland. We do not have as much white, indicating that there's much more water. And here on the parotid gland, we got a good cross-section of that secretory end piece, or one of the secretory end pieces, so you could see the lumen on the inside. And like I said, this is secretory end piece is interchangeable with acini. Acini is plural. Acinar is singular. So if you have one, it's acinar. So this is one acinar, or secretory end piece. So next we're going to discuss some of the structures and histology of the rumen. So we got into it a little bit within the first lecture, but we're going to get into much greater detail here in this lecture. So in the last lecture, I showed you one picture of the rumen. So this is a picture, a schematic of the rumen on what we'd see on the left side view, and I'm sorry for the poor graphics here, but it was the best I could find of the left-hand side. So here you could see left-hand side, the food comes into the esophagus and it goes into the reticulum, and this whole big component here is the rumen. And you can see within the rumen, even though it's a large sac, we have different compartments within it, and they all have different functions. So we have the cranial sac, the dorsal sac, and the ventral sac. In addition, we have this reticular omasal orifice here, and this is the component that's going to lead the ingesta to the omasum, and we have this esophageal groove. So interestingly, this esophageal groove is evolved because when a young calf is first born and its main diet is consuming its mother's milk, Mother Nature needed to ensure that milk did not go into the rumen where it could be fermented and it caused a lot of gas pain. If anybody is lactose intolerant, you're familiar with cramps, etc., bloating that happens due to fermentation of any dairy products that you are unable to digest. So these animals evolved to have this esophageal groove. So once the animals, by the way they stand, if you ever watch a calf nurse, it has its legs pretty far out. It almost, and it tucks its head under its mother's abdomen to nurse. And when it puts its head up that way in that stance, it actually causes this esophageal groove, those two flaps of tissue that are actually able to bypass the rumen, so that milk is able to bypass the rumen and go straight into the omasum and abomasum, because milk is one of the most nutritionally complete foods. The calf does not need assistance from the microbes to break all of that down like it will later in life when it is reliant solely on those microbes to meet the majority of its energy needs. So here's the same picture that we had in the last lecture. So this is looking at the right side of the animal, what you see. So you have the esophagus coming in. So the feed will enter the rumens. The rumens, that big vat, different sacs within the rumen, the reticulum, and then we have the omasum and the abomasum. So the reticulum is, by all intensive purposes, the first component within the ruminant stomach. So it traps any non-feed stuff. So as I said in the last lecture, a lot of dairy farmers, cattle ranchers, they will actually put in magnets, and those magnets stay within the reticulum, and that's to catch any sort of metal that the animal may consume that we don't want to have puncture later on in throughout the rest of the GI tract. And you can see that the tissue, it's mucous membrane, and if you look at it, it actually has these honeycomb ridges here. So it almost looks like a honeycombs that you would find from that bees develop. The lining of the rumen, reticulum, and omasum, all of them, even though if you look within these three different sections of the stomach, they all look very different. So the reticulum has the honeycomb. If you look inside the rumen, it almost looks like seaweed with these very elongated papillae. And the omasum almost looks like leaves. But all of them are composed of, all the structures are composed of papillae or leaves, which are finger-like projections in the rumen and omasum. And all of these papillae are what we refer to as stratified squamous epithelium. So that means it's very similar to the skin. So you have the layers of skin, the cells are on top of each other. So that is what stratified squamous epithelium means, which is different than once we get into the intestinal tract, then those are columnar epithelium, where there's just one single layer. And we'll discuss that a little later. So here we have some pictures of the, you can see here on the right-hand side, the reticulum of a sheep, that honeycomb pattern. Those are also, remember, keep in mind, made up of papillae, even though it looks completely different than the papillae found within the rumen. And then we have the ruminal tissue. Again, it almost, if you were to look inside a rumen, it reminds me of seaweed. If you're watching seaweed move in the ocean back and forth with the fluid. So here's another picture comparing the rumen papillae versus the reticulum papillae. Reticulum papillae, honeycomb, rumen papillae, looks like leaves, seaweed, moving together. Now, the integrity of these rumen papillae are very critical for the health of the animal. So most beef cattle in the U.S. are actually what we refer to as finished at a feedlot, where we give them a significant amount of concentrate. So if you recall, cattle have evolved to consume mainly grasses, and they're referred to as grazers. They eat various grasses. And if you continue to feed cattle or sheep, hey, they have these very healthy papillae. However, once you convert them, and a lot of times these finishing diets are about 80% concentrate, which is corn and other various grains, high starch that are easily fermentable, and the animal grows really efficiently off of those. However, you can see from the schematic how that alters the papillae. So these papillae end up becoming very frail and brittle, and that's because of the microbes that are developed with it that are able to utilize that, and they change the volatile fatty acid. And therefore, that change in volatile fatty acids that's produced affects the pH of the rumen, which inadvertently ends up deforming the papillae. The third section of the stomach is the omasum, and this also is made up of the stratified squamous epithelium. However, you can see with this cross-section, a schematic of the omasum, we have, it almost looks like leaves. So this is very important for water and volatile fatty acid absorption. It also is used as a filter because we want to make sure that anything that's going through there is a certain particle size. So the omasum is filtering everything out. It's absorbing water and volatile fatty acids. So within the rumen, this stratified squamous epithelium, we actually, and this is pretty similar to skin, we have different layers. And the first layer is referred to as the stratum basal. So this is fully metabolically active, live. These are what we would refer to as the stem cells. They're capable of mitosis, so they are constantly proliferating to maintain the epithelium, the integrity of these papillae. The next layer, the stratum basal, is also the one closest to the blood, so they have the first access to all the nutrients. The next layer is the stratum spinosum, and this is where cells begin to lose some metabolic activity, and they develop some spider-like processes. They're still alive, but where they're less metabolically active. So they are further away from the blood supply. The nutrients are diffusing through, so there's just not as much for them to get. Then we have the stratum granulosum. And within this layer, the keratin bundles begin to form in the cytoplasm. So keratin is a type of protein. The tight junctions between the cells start to become leaky. So you have selective passage of some substances. The cells are still alive, but just barely. They're the ones that are on life support. And then you have the stratum corneum, and these cells are the ones, they are completely dead. So they are the ones that slough off. They're similar to the top layer of skin. So if you ever have dandruff or whatnot, the skin cells are able to slough off, that you're not injured. So the stratum corneum, exactly the same. And this ends up allowing for a particular micro environment for some facultative rumen bacteria that inhabitate the rumen. So we have a nice symbiotic relationship that ends up occurring. So here is the picture schematic of this layer. So you'll see at the in the middle we have this stratum basal. So these cells, here we have our basement membrane, basal membrane, and where the blood's able to come through. The basal cells, these are fully alive. Then we move into the spinosum. So you can see there's still some room where these nutrients can diffuse through and keep them alive. Granulosum, you can see the nuclear membrane is starting to get lost. We have some leakiness, and eventually we lead to the corneum, where we no longer have the nuclear membrane, and the cells are completely dead, metabolically inactive. Okay, next we're going to discuss the structure and histology of the esophagus, stomach, also referred to as the gastric pouch. So after we leave the oral cavity, the food moves down and it first enters the esophagus. So this is the part where we transfer the ingesta, because once food is in your mouth, we refer to it as ingesta. So it's the passageway from the mouth to the stomach, or directly to the intestines for animals that lack stomachs. It also serves as a temporary storage site for some animals, primarily being avian species. There are many avian species that have this pouch within the esophagus, and that component is referred to as the crop. So they are able to do some food storage there. It's not a significant amount of food storage, but it can help them overnight fast, etc. And the esophagus is a non-glandular stratified squamous epithelium. So there isn't any glands, there's no secretions, there's no saliva being secreted. Some species do have goblet cells, and these goblet cells actually secrete mucus, but goblet cells are not glandular. So some species do have goblet cells, but there are no glands. And this also, that stratified squamous epithelium, similar to how the skin is. So here's a little video showing peristalsis moving down into the, this is actually within the stomach, so a person who had an endoscope done, and you can see the how the everything is moving through. So food moves through the esophagus with peristalsis, and I just thought this was a really neat video. Clean stomach, showing how all the muscles are contracting and moving everything through. So within the stomach, we also have different compartments. So we're going to start off with the human stomach. And we have, in the human stomach, we have the esophagus, leads into the stomach, and we have different compartments within the stomach. So this first part near the esophagus is referred to as the cardia, mainly because that's closest to the heart. You have the body of the stomach, which includes the fundus and the antrum, and then the distal part of the stomach is referred to as the pylorus. We do have the pylorus sphincter, which is controlling what is actually secreted into the first component of the small intestine. And then we do have reverse peristalsis that can occur within the stomach. If anybody's been subjected to heartburn, so that is where the gastric contents are able to regurgitate, and they end up causing a burning sensation in your esophagus, and that's due to the acid having its effect on the squamous epithelium, and the stomach has a very low pH, so basically you're burning your esophagus. In addition, anybody who has encountered or had the experience of vomiting, that's also what is occurring. Not all stomachs are the same. So within the stomach, depending on the animal, the functions vary. So here you can see on the left-hand side with the human, we have this first component closest to the esophagus is the stratified squamous region. So in the human, that's very, very small. However, if you compare that to the horse, which has significant amount, so basically there, it's mainly for storage. And you can see how similar the human and the dog stomachs are. And the next region you have with this slightly more dense dotted area is the cardiac gland region. So here it's pretty small in the human and the dog. It's pretty significant in the pig. And this is where the animal is actually secreting the mucus. And we need to secrete that mucus because we want to protect the lining of the stomach, because the stomach does have a significant amount of hydrochloric acid that is released to help instigate the digestion of food. So we need to protect the stomach. So we need to secrete mucus. If you don't have adequate mucus, you'll end up getting what we refer to as ulcers, where you'll have that hydrochloric acid burning holes into the stomach. The next component is the proper gland region. So this is the dark black area. So you can see it's significant within the human and the dog, less prominent in the pig and the horse. And here in the ox, this is the abomasum component. You can see in the rat, less prominent than we have in the human or the dog. And this is where the gastric juices are secreted. So this is where we have the secretion of pepsinogen and hydrochloric acid. And then the last region, the striped region, here this is going to be the pyloric gland region, and this is secreting gastrin, the hormone gastrin, which is controlling gastric emptying and any mucus. Within the stomach, you also have these folds called rugae. So here we have, this is a human stomach that's been opened up, and you can see all these little folds here, and those are there because they help increase the surface area, and they're also what's allowing the stomach to expand. So everybody just got back from Christmas break, having holiday food, and so many times you end up overeating. So without these rugae, we would not be able to cram as much delicious holiday food in us as we would without them. So they're increasing the surface area, also what allows the expansion of the stomach. So within the stomach, we have different types of gastric glands. The first one, as I kind of mentioned earlier, is the mucus cells or mucosal cells. And these are the cells that are secreting the protective mucus coating. Early within the cardia region, so we want to secrete the mucus to protect the lining of the stomach. And the next set of cells we refer to as chief cells. And these chief cells are secreting the zymogen, pepsinogen. So a zymogen is an enzyme that is inactive when it's first released. So chief cells secrete the zymogen pepsinogen, And then we have parietal cells. They secrete hydrochloric acid and intrinsic factor. So the hydrochloric acid is there to help denature many of the proteins. It also ends up activating pepsinogen to pepsin. And intrinsic factor is a protein that is released that you need to bind B12 so you can absorb B12, and we will talk about that much later in the semester. So here we have another schematic of the gastric glands. So here, these mucus cells are going to be more at the top. The chief cells that are secreting the pepsinogen, they are going to be down within these cryptic areas. Parietal cells are more on the stalk, and then the mucus is up at the top layer. Okay, next we are going to get into discussing the small intestine. So the small intestine can be split up to three different components. There are three sections of the small intestine. The first component is the duodenum or duodenum, depending on where in the country you come from. I grew up with, we call it duodenum. So it makes up about 15% of the small intestine. There's very little absorption that occurs within the duodenum, and it's very rich in endocrine cells and receptors. The next section is the jejunum, and this is where the majority of absorption is occurring in most species. So the duodenum is where you have the secretion of the enzymes. Digestion is mainly occurring there. Absorption is mainly occurring in the jejunum. And the ileum is the final segment of the GI tract before we get to the cecum or the colon. And after the jejunum, it's the most important site of absorption. And interestingly, if you ever known anybody that, for instance, has had Crohn's disease and therefore have had to have sections of their intestines removed due to abundance of inflammation that occurs, if that individual has a significant amount of the jejunum removed from the intestine, the ileum can actually pick up and take over a lot of the absorption that the jejunum did. However, the reverse is not the same. So the ileum, if it has receptors and sees that there's a significant amount of amino acids, glucose, etc., it will be able to pick up the slack and increase the absorption, where if you have a significant amount of the ileum removed, the jejunum will not know to do the functions of the ileum. So we have different levels of organization within the small intestine. So if you were to just look at the intestine on the outside, it's a basic cylinder. However, we also end up having a significant amount of folds within the small intestine, and then we have within those folds, which is similar to the rugae, we have the villi, and then we have microvilli. So all of this ends up affecting or increasing the surface area of the small intestine. So if we were to have just a cylinder, we would have an amplification factor of 1. And then when we add in the folds found within the small intestine, that ends up increasing the amount of the surface area by 3 times. And then finally, within these folds, we have these villi. that increases the surface area 10 times. And the cells within those microvilli also have, or those villi are made up of the enterocytes, and they have these microvilli, which almost looks like hair. And that ends up increasing the surface area by 20 times. And I think I really want you to, you don't have to tell me the centimeters of the surface area for the human, but I do want you to be able to tell me the amplification factor of the different levels. So we also have the structure of the small intestine. So within, we have the serosa, which is green here. The serosa is the connective tissue keeping everything within the intestine. And then we have two muscle layers. We have an outer layer and an inner layer. So the outer layer has this longitudinal component, and the inner layer has this, it's more circular. So we need these two layers for peristalsis. So the first component is pushing things down. So the outer layer is pushing things down, and then we have the inner layer, which is actually bringing things in, and that combination is what's really helping with the peristalsis and moving everything out. And then we have the submucosal layer followed by the mucosa. And we also have the mesentery, so this is connected to the serosa. It's another component of the connective tissue, which is keeping everything in place within your abdominal cavity. So here we have a magnified view of the small intestine. This is a human. So it's very difficult. You can't see the microvilli, but all this fuzzy component is actually the microvilli. So here we would have what would be the cylinder component. You could see these folds within, and on these folds we have these villi. So all of that is what's increasing the amount of surface area on top of those villi. Each one of those cells has what we refer to as microvilli, which is increasing the surface area even more, and that allows us to absorb even more of the nutrients. This is a picture on the right-hand side of the jejunum of a broiler chicken. Broiler chickens are the ones that we actually buy the chicken carcasses in the store. So here you can see the jejunal villi of this chicken. And this is a electron scanning microscope, so you get a nice 3D image. So you can see the finger-like projections, and these vary in shape depending on the species. You can see all the bacteria, all these little lines. Those are different bacteria that are attached to the intestine. They're embedded into the enterocyte. So the enterocyte is the absorptive cell within the small intestine. And you can actually find within neonatal animals and avian species, so very early, the intestine starts getting colonized by this bacteria. And actually they found in some fetuses that they start to have some colonization occur there. So it goes against what most people have always thought about as far as when bacteria start colonizing the intestine, that it actually starts before an animal is born or hatched. So we also have different parts of the villus. The lower part is what we refer to as the crypts of Lieberkron, and these are located at the base of the villus stalk. And this is where all the new enterocytes, the goblet cells, and the endocrine cells reside. And as the enterocytes mature, they migrate up the villus stalk. And you end up having, there are multiple crypts that are associated with each villus. And next we have the villus stalk. So these are covered with enterocytes, which are the absorptive cells. They are not fully mature when they are at the villus stalk. We also have goblet cells reside there, and as I mentioned before, goblet cells are the mucus-secreting cells. They are rest on the basement membrane. And then we have the internal core. So this internal core is where we have all the arteries and veins and the lacteals. Lacteals are the same as lymph. All of those are located within the core of each villus. And all except poultry. Poultry do not have any lacteals. So poultry have a very undeveloped or underdeveloped lymphatic system. So they do not utilize the lymphatic system to absorb fats like many mammalian species do. So, very important to know, poultry do not have lacteals or lymphatics throughout the core of the villi. That may be a question on the exam. So here on the left-hand side, we have another schematic of what it looks like. So here we know that this is not a schematic of a chicken because we have an artery, vein, and the lymphatic system going into the villus. So a chicken does not have the lymphatics. So we do have arteries, the veins, and lymphatics. Here we have the crypts of Lieberkron, the villus stalk, and these little white things would be our goblet cells secreting mucus. And here is a histology example. So here we have at the bottom here the crypt, and this is where we have all the enterocytes, endocrine cells, and the goblet cells first form, and they move up the villus stalk. And here we have, at the top, we have the enterocyte layer. So you can see, you can also see here that the difference that these cells are actually columnar. So all the nuclei at the bottom of the basement membrane. So all those little dots indicate a cell. They're all close together. So we have the villus stalk up top. And then on the inside you can see this is where all the blood cells are with all the arteries and veins that go into the villus. The white components here are the goblet cells. So the mucus does not take up this particular stain very well. So all those white blank spots are actually your goblet cells releasing mucus. Here's another picture of it. So here we can see the veins, arteries, and lacteals within the villus stalk. Here with this blue, we have mucus being released out of a goblet cell. We have the enterocytes over here in between these goblet cells. So this blue is this stain actually was taken up by the mucus protein. So that's why you can see the blue. So I've mentioned a few times about enterocytes. So enterocytes are the absorptive cells within the intestine, and they cover the villi. And they are produced in the crypts, and they are fully differentiated, meaning that they are fully functioning, mature by the time they reach the mid-villus. The exception is the chicken. So chicken can actually have the enterocyte mitosis occurring at the tip of the villi. So chickens are the anomaly. These cells, when you look at them, they are quite polarized, where you can see the nucleus is always located on the basolateral side, and the microvilli brush border are always on the other side. And this is very common, so we refer to these cells as columnar epithelium because they make a column, they're single layer, and they rest on the basement membrane, separating them from the underlying serosa and muscle. So here's some slides of the chicken enterocytes. So here you can see these dark components here. Those are actually enterocytes dividing at the villus tip in the chicken. Currently, the chicken's the only one that we've observed that can do that, but you can kind of see here these all the nuclei are all lined up. I have a better picture next. Here is the picture, electron scanning microscope photo of the mucus blanket. So those goblet cells are secreting mucus, which provides a nice protective layer for the intestine. And there are many benefits. It slows down absorption, which helps regulate various diseases, such as if you have too much glucose absorbed right away, you're going to be more prone to develop diabetes. So this actually is good to have this mucus blanket here. And with this picture, you can see on the left-hand side, there isn't a mucus blanket, but on the right-hand side, you can see the mucus nicely covering the villi. Here's the electron microscope photo of the intestine of a villi. So here you can see the nucleus, the nuclei of all these cells. We have the basolateral membrane at the bottom, and you can kind of see where each enterocyte is. So each E is with the arrows, that's where we have the enterocytes. And you can see we have these very nice tight junctions. So we want those very little space. We don't want anything to be able to sneak past the enterocyte, which would include pathogens. We don't want, so we have, these cells are very close together. And you can see the microvilli, that very dense area, so that's where the microvilli are and all the absorption is occurring. Okay, next we're going to move on to the pancreas. Pancreas is very critical for the GI tract. It's usually referred to as an accessory organ because it's not directly part of the cylinder from the mouth to the anus. But the pancreas is what is providing the majority of the initial enzymes to break down the food to so it can be better absorbed. So in both the human and the chicken, the pancreas is very easy to find. It's lobular. It's found at the beginning of the duodenum. In the chicken, it's the component. There's a loop of the intestine called the duodenal loop, and the pancreas is the organ that lies in the middle of that. So it's very easy to find in both humans and chickens. However, some other species like the rat, it's much more diffuse and spread throughout the mesentery, or the connective tissue next to the duodenum. So it really depends on how clear, depending on the species, where the pancreas is. So as I mentioned before, the pancreas is what secretes, it secretes buffering fluids. So remember, we have that hydrochloric acid coming from the stomach, which is a very low pH, and if we don't buffer that and bring it up to a more neutral physiological pH, it will have very bad ramifications on the rest of the GI tract. So we need to secrete some buffering agents, and it's also very rich in various digestive enzymes. The pancreas has multiple ducts that empty into the small intestine. The chicken, for instance, has 3 ducts that empties into the duodenum, where humans and a lot of other mammals have only 2. The basic secretory unit has the alveolus within the pancreas, so it's kind of similar to the salivary glands, those acini or secretory end pieces. So the secretory unit, it looks like that. And within the pancreas, it's not only secreting digestive enzymes, but it also is a major endocrine organ. So we have different components within the pancreas that releases insulin, glucagon, and which help control glucose, regulate glucose. And these are secreted, and we have clusters of cells called the islets of Langerhans. So these different components, all these endocrine, are clustered together called islets. So if you know anybody that has type 1 diabetes, they typically don't have these islets. There's type 1 diabetes is autoimmune, where your body attacks the beta cells within the islets, so they no longer secrete insulin. So here we have an example of the small intestine, the duodenum, and the pancreas. So this came from a human. We know that, one, because the pancreas is very identifiable, and there are 2 ducts, and these open independently, and the ventral one opens out into the common bile duct here, and we have the ventral pancreatic duct. So these are able to, the enzymes or pancreatic juices are able to be secreted into the ducts, and they're released into the duodenum. So this is looking at the histology of the pancreas. So the dark blue staining is the acinar cells. So these are the cells that are secreting the enzymes, the zymogens, that are going to be utilized to help break down various feeds and feedstuffs. The black dots are the nuclei of all the cells, And then this light staining here is what we refer to as the islet of Langerhans. So all these cells, this light blue staining, is actually the endocrine cells that are secreting insulin and glucagon. So this is what the alveolus looks like. So it's pretty similar to the salivary glands. So we have the acinar cells here that make up the alveolus, lined with the secretory cells. So they all secrete and empty into the lumen, and then the contents within the lumen are actually able to leave out of the ducts. And this, as I said, very similar to salivary glands and the mammary glands. The next, we're going to talk about the gallbladder. So the gallbladder is critical for proper lipid digestion. So the gallbladder, in species that have them, it lies under the liver. So here we have a schematic of the liver. So it's right underneath. So this lobe, if you were to pick up this lobe, you would see this emerald green sac. That is your gallbladder. So the main function of the gallbladder is it secretes bile for the emulsion of fats. So you need bile to properly absorb fats, and depending on the species, you'll have, some species don't have gallbladders, and it all depends on what they are eating. For instance, horses don't have gallbladders. They're mainly eating grasses that don't have a lot of fat. In addition, horses have evolved to eat constantly. They should eat. Naturally, they eat about 15 hours a day. So since they're constantly eating, their bile is secreted straight from the liver. But for other animals, such as humans and cats, etc., that have more meals and are not eating constantly, we have a gallbladder that upon eating, we can release the gallbladder and have adequate bile to digest the lipids. So here's a liver from a human, here from a cadaver, and you can see this emerald green. This is the gallbladder. So this is the under the liver. So if we were to flip it, it would be very difficult to see the liver when you first open up a body. But if you pick up this lobe, you'll find that the gallbladder right underneath. Next, we have the colon and cecum. So this is the site of lower gastrointestinal fermentation. This is where we exchange water and electrolytes. We store digesta. There's very little absorption of energy-yielding nutrients, so we don't have direct absorption of glucose and fructose, lipids, etc. This is where we're going to be mainly absorbing water and electrolytes. And importantly, the colon and cecum, they only have crypts from which we secrete mucus. They do not have any villi. So here we have a schematic of the colon, so you can see the significant amount of goblet cells found within the crypts of the colon. And we still have the arteries and veins and lacteals found within these crypts. But once again, notice we don't have any villi. They're just crypts. So this is a histology of the colon. You can see just the crypts, a significant amount of goblet cells that are found within the colon. So the goblet cells are the ones with all the white, so a lot more goblet cells than what we saw in the jejunum or the ileum. So this is a ultrastructure of the colon by an electron microscope. So basically here you can see the mucus secreting cells within the colon here. And then finally, this is an endoscope of an individual getting checked for colon cancer. This is our public service announcement. So want to remind everybody that colon cancer is the second leading cause of death in both men and women. And once it gets beyond its early stages, it's extremely aggressive. So it's very critical for people to go and get colonoscopies. Usually, colon cancer develops very, very slowly. Usually you'll have some sort of fibroid that initially is there that isn't giving you much problems, and then or you get these polyps. The fibroids develop into polyps, and then that's basically where you have the colon cancer stem from. So if you regularly go and get a colonoscopy, you can discover any of these polyps and prevent any colon cancer to having them develop into cancer. So it's very easy to, you don't need any treatment if you are able to have all the polyps removed, but once those polyps are transformed and become cancerous, they're very aggressive, and that's why we have greater, that's why it's the second leading cause of death, and most people don't think to go in until they're having many complaints on gastrointestinal issues, and at that point in time it's too late. So after you're 50, I believe it's once you're 40 they want one. Doctors will start prescribing them at 40, but after 50 you need to have one done every 5 to 10 years, depending on your family history.